But now what I'd like to do is to turn our attention to the large-scale events that account for the structure of the adult brain as now you've come to understand it as we've worked through the early units of this course. So, how is it that we can go from a simple tube to this marvelous structure that you've now come to appreciate as the adult form of the human brain? It's hard to look at this, is it not, and imagine that at some early stage in development this brain was a simple tube, but indeed that's the case. So what I'd like for you to be able to do is to reflect upon what you know about the anatomy of the adult form of the human brain and relate it back to the more simpler forms that are produced in embryonic life. Okay, well, to get there, let's just look at the development of the brain and the spinal cord in the very broadest terms, as if we were looking at this from a fairly macroscopic perspective. Well, what I'd like to show you are a series of illustrations that show the various transformations of regions of that neural tube that give rise to specific parts of that adult human brain that you now know something about. Well, over to the right we have an illustration in schematic form of our neural tube, and so it's a fairly simple representation of that tube, but we're really only showing the anterior end of that tube. To the left we have a picture of the developing embryo. So here is the embryo at a fairly early stage in development, probably about 4 weeks or so, 5 weeks, and what we find here is that the developing embryo has a shape that looks something like a cane, as our textbook describes. I hope you're not offended by that. This is vertebrate life, after all, and a representation of human life. But what we find is a shape that reflects some morphogenic events that begin to take place in the anterior end of this developing neural tube. What I mean by morphogenic events is the differential proliferation of cells that cause this simple neural tube to begin to bulge in certain places, to bend in others, or to constrict. And what we find is the formation of essentially 3 principal enlargements in the anterior end of this neural tube. The most anterior of these enlargements we call the prosencephalon. So the prosencephalon can be found here at the anterior end of the neural tube, and if we see this cane-like shape, it's at the end of the handle of the cane, so right in this region. And this prosencephalon is what will go on to form the cerebrum, the hemispheres of the brain. So it's the front brain, the first brain, that's why we call it the prosencephalon. So just behind the prosencephalon, we have the middle of these 3 swellings, and we call that the mesencephalon, which means middle brain. So this middle brain will go on to form what we call the midbrain. And then lastly, the third of these swellings, just caudal or towards the tail, behind the mesencephalon, is called the rhombencephalon. And the rhombencephalon is what we call the hindbrain. So the rhombencephalon would be this relatively extended straight part at the top of the cane-like shape of this developing embryo. And then caudal or posterior to the rhombencephalon, we have the spinal cord, which will take shape. So these 3 principal divisions go on in development, and they begin to further subdivide, specifically the prosencephalon and the rhombencephalon. So the prosencephalon begins to divide into 2 principal divisions, a diencephalon and a telencephalon. The telencephalon will begin to form these large vesicles that will continue to expand and dominate the developing nervous system. So these telencephalic vesicles are what will give rise to the cerebral hemispheres. The diencephalon, meanwhile, undergoes its own interesting changes. The diencephalon will begin to form these outpockets that we call the optic vesicles. And the optic vesicle is what will go out to form the neural elements of the eye, specifically the retina. And of course, as I mentioned when we study the visual system, the retina can therefore be seen as really a part of the brain. And the connection between the retina and the rest of the diencephalon really reflects its embryological history. This is why the optic nerve is myelinated by oligodendrocytes, the glial cell that makes myelin in the central nervous system. And this is why, in one perspective, we might argue that the optic nerve really isn't a nerve at all. It's an extension of the brain. Okay, well, let's move on and consider briefly what happens to the mesencephalon. So the mesencephalon is that middle brain. This is the division of the developing neural tube that seems to not change so much relative to the others. It doesn't necessarily increase in size proportionately, nor does it differentiate or subdivide any further in any significant way. So just caudal to that mesencephalon then is the hindbrain, the rhombencephalon. And the rhombencephalon actually does begin to show some interesting differentiations. There begins to be a bend at the top of that cane-like handle, and that we call the pontine flexure. And this flexure begins to differentiate two principal components of the rhombencephalon. In the more rostral direction, we have a metencephalon, and then more caudally, just posterior to this flexure, is the myelencephalon. So the metencephalon is what will go on to form the pons and the entire cerebellum. The myelencephalon will form the medulla oblongata. So let's advance one more stage in embryogenesis. So now we're well into the second trimester of gestation, and we begin to see a nervous system that resembles what you've come to appreciate in the adult form. So the view of the developing brain at this point is obviously dominated by the cerebral hemispheres. So this is, of course, the telencephalon. And if we were to look at this forebrain in cross-section, what we would see is not only an outer cortex developing in this telencephalic vesicle, but also we would see deep gray matter. And that deep gray matter, of course, is the basal ganglia. Well, not seen from the outside, but present and visible in cross-section, is that derivative of the prosencephalon that is immediately caudal to the telencephalon. And what I'm referring to, of course, is the diencephalon. And that diencephalon is beginning to differentiate into a thalamus, which is found more dorsal, and then not seen at this level, more ventral and a little bit anterior would be the hypothalamus. So from the diencephalon, we get the thalamus and the hypothalamus. And then if we looked a little bit deeper, also not shown in this particular cross-section, we would see the mesencephalon, which is now completely dwarfed by the expansion of the prosencephalon into the telencephalon and the diencephalon. And just caudal to that mesencephalon, we would see next the derivatives of the rhombencephalon, or the hindbrain. And those derivatives would be most obviously the cerebellum, and that portion of the brainstem to which it's most intimately connected, and that would be the pons. Together, the pons and the cerebellum then are the metencephalon. And then we would begin to see a nicely shaped myelencephalon, which is a transitional region between divisions of the brainstem and the upper segments of the spinal cord, which is, of course, what then extends down the vertebral column of the developing fetus. Well, what I'd like for you to be able to do is to tell this story of the major events that shape the formation of the brain, and to be able to relate the adult brain structures that you've now come to recognize and know something about their structure and their physiology. I want you to be able to relate those derivatives back to the embryonic brain. And I want you to include in that discussion the associated ventricular spaces. So hopefully, as we begin to put together your understanding of the anatomy of the human nervous system, this embryological framework will actually come to help you. So for example, if you are looking at a cross-section through the brain and you're not quite sure what you're looking at, perhaps you will remember that what you're seeing near the dorsal aspect of the cross-section is actually a very narrow channel, allowing for the flow of cerebral spinal fluid from the third to the fourth ventricle. You may recall that that's the cerebral aqueduct, and that would be a hallmark of the midbrain. So seeing the cerebral aqueduct is one way to identify what kind of section through the brainstem you might be inspecting. Well, having an understanding of embryogenesis helps you appreciate why the lateral ventricles show up where they do. The lateral ventricles are present both along the dorsal aspect of the brain, but also down into the temporal lobe and even extending back into the occipital lobe. And the development of the temporal lobe is an important aspect of human brain development. And I think what you have come to appreciate is that there are certain structures in addition to the lateral ventricle, such as the caudate nucleus, a component of the basal ganglia, that are pulled down and anteriorly into that developing temporal lobe. So having this embryological framework, knowing where things came from, can be a great aid to you in understanding your appreciation, your perspective of human brain anatomy. Well, if you've not done so yet, I would encourage you to view my brief tutorial on the embryological divisions of the human brain, and to use this chart as a way to reflect upon what you're seeing, as that video tutorial brings you back to the lab and shows you the human brain and allows you to recognize the various subdivisions that I've been talking about as they actually present in an adult brain specimen. So if you've not done so, go ahead and do that at this point, and we'll resume our discussion in this tutorial after you've had a bit of reinforcement of your ability to recognize human brain anatomy in the adult form and be able to relate that back to the embryological origins.